This is note taker 14 on genetic engineering. Probably one of the most common types of genetic engineering is going to be where you take DNA from one organism and you add it to the DNA strands of a totally different organism. This can happen because every living thing in the world, their DNA is made up of A's, T's, C's, and G's. Now, cloning is also a type of genetic engineering. We'll be talking about that later in class. The type of genetic engineering I'm going to concentrate on this note taker, though, is going to be the part where the DNA from one organism is mixed into the DNA of another. And we call this new DNA that's created recombinant DNA because it is recombined from two types of organisms. Now, the one I'm going to show you in a picture on here is going to have to do with bacteria and humans. So we're going to take human DNA and we're going to take a piece of it and we're going to actually put it into the DNA of bacteria so that bacteria, which produces more quickly, can actually make chemicals that the normal human body would make. But it'll make it a lot faster than our human body can do that. When we make recombinant DNA, we're going to need DNA from two different living things. The example that I'm going to show you here and then on the next slide in a picture, we're going to take a little tiny piece of human DNA that codes for a human growth hormone. We're going to take this DNA and we're going to put it into the DNA of a bacteria. So as the bacteria reproduces and makes more bacteria, all of these new bacteria will make this growth hormone for us. And because bacteria reproduces much more quickly, it can produce a way larger amount of human protein than our bodies can actually produce on our own. So, the reason why bacteria is used, because it's very simple. There's one strand of DNA and it's in the shape of a circle, and we call that ring a plasmid. So what scientists are going to do using their special tools, they're going to take this plasmid and they're going to make a tiny little slice in it so that it opens up the ring. Then they're going to take a section of DNA from a human cell that codes for the growth hormone, and they're going to take this piece and where they made the cut in the bacteria DNA, they're going to insert the human DNA in there. And then the chemical bonds will rejoin and you will now have a new piece of DNA that's going to be put back into the bacteria that still has all the bacteria DNA, but also now has a little chunk of human DNA. On the next slide, I will show you a visual or a picture of this so you can see and understand how this happens. Here in this picture, this is making recombinant DNA. The top left-hand corner shows a human cell, and next to it has a little arrow pointing to the right, and we have a little gene for human growth hormones. This is a tiny little section of human DNA. On the bottom left, it shows the bacterial cell, and that little ring at the bottom is the plasmid, which is its DNA. So we're going to take that out, we're going to cut it so that that circle opens up, and in the top right picture you see where the little piece of purple human DNA has been put into the bacteria DNA, and then that will be put back into the bacteria as shown in the bottom right picture. Now that bacteria will actually produce a human growth hormone. And because bacteria reproduces so quickly, every time that bacteria splits to make new bacteria, each one of those new bacteria will now carry that human DNA and will also be able to make human growth hormone. Now, making recombinant DNA sounds very easy, and actually, if you have the right tools, it is pretty easy, but it's not something that you can run home and do in your basement. You're not going to be able to have all these weird creatures running around. But why we do genetic engineering The good reasons are to help us in both medicine and agriculture. Those are two of our good examples. In medicine, we look at being able to produce these human proteins, and we can actually use these proteins to make tests for other diseases. We can also use them to make vaccines to fight off diseases that we can get, such as hepatitis B. Or we can try to treat certain diabetes or certain disorders like diabetes. Now, when we talk about diabetes, this is a disorder that is genetic. So there is something in your code that tells your body not to produce insulin the correct way. So people who are diabetic, a lot of them need to take insulin. Now back in the olden days, if your body's not producing enough insulin, you have to inject it somehow. In the olden days, they would use insulin from cows or horses. The only problem is that's not exactly the same as human DNA, and people had reactions to it, and it didn't always work really great. So by using genetic engineering, we've been able to use bacteria to produce human insulin for us, so that people who are diabetic can get plenty of insulin so that they can maintain their sugar levels. Now, when we look on the plus side for agriculture, we've actually been able to take different mutations over time that have made some plants resistant to certain things, and we can take that gene code and put it into other plants. So now we can make plants that are resistant to the cold, resistant to drought, which is no rain, and resistant to some diseases. So these are good ways that genetic engineering is used. Now on this note taker, I concentrated just on the making of recombinant DNA, which is just one topic in genetic engineering. However, there are other topics we will be discussing in class, and over the next couple classes we're going to be breaking into groups and doing some research on these topics. The topics that we'll be discussing in class will be cloning. We're going to talk about how cloning works and some ethical issues behind it. We're going to be talking about GMOs, and in class you'll learn what those are. We're going to talk about selective breeding. We're going to talk a little bit more in depth about genetic engineering, the gene splicing, which is the recombinant DNA. We're going to talk a little bit about gene therapy, and then we're going to talk about stem cells. So these are all topics that we'll be researching over the next couple of days in class. I tried to show you a couple of goods with genetic engineering, how we use it for medicine and how we use it for agriculture. And then over the next couple of days, as we're researching these other topics, you're going to find that there are some goods and there are some questions, some ethical questions. So when we talk about genetic engineering, we need to always keep in mind, when does genetic engineering become too much? Or does it ever? And that's going to be based on personal opinion. But I will show you some movie clips in class, just some little brief movie clips about things that have to do with genetic engineering. And we'll talk about the ethical issues behind that and whether or not you think these ideas are good or they're bad. Please don't forget to complete the assessment that follows the video.